So our final talk for this morning's session is a second plenary talk, and that's Next Generation Immunogenomics to Understand Health and Disease from Professor Muzlifah Haniffa, Wellcome Trust Senior Research Fellow in Clinical Science, Professor of Dermatology and Immunology in Newcastle. You're most welcome. We're delighted to have you with us. Thank you.
Thank you very much, Alan, for the introduction, and it's fantastic to be here. One of the first live meetings that I've been to after the pandemic. So I've been given this talk, and I thought I would give a little bit about the science that has been happening in the last few years, but also a little bit of the personal journey behind the science. So like many of you, I was a dermatology trainee undertaking research, and my PhD project was essentially focused on dissecting what were the antigen-presenting cells that were in the human skin. And that was funded by an Action Research Training Fellowship. And thereafter, I went on to a clinical intermediate fellowship to then compare the population of antigen-presenting cells in the human and mouse skin to try and align the populations. And I kind of want to just, show the next slide in terms of my career is not just about going from one fellowship to another. I mean, this kind of idea of a linear career trajectory is not necessarily how it works, but sometimes it's just worth applying for these fellowships, because when I applied, the interview was 10 weeks after I gave birth to my first son. So going for an interview at that stage, I remember being in the train and thinking, what am I doing? But I arrived at Wellcome Collection, and there was this fantastic exhibition of a woman who drew every day of her life through mental health illness and came out of it throughout her treatment. And I was so inspired, it helped my interview. And then I went to a senior fellowship, and again this time I thought I had this great idea. I'm going to leverage this cutting-edge technology that has just come about using single-cell RNA sequencing to really define the heterogeneity of mononuclear phagocytes, antigen-presenting cells. And this time I was going to choose blood. So the concept is very simple. You have a mixture of cells in a tissue, which if you did bulk RNA sequencing, you won't be able to dissect the individual cell types. But by using single-cell genomics, you can actually deconstruct the tissue to its cellular constituents. And then I found that actually, the project that I had outlined was essentially being done by a group at the Broad Institute. So I just reached out to them and said, I'd quite like to work with you. And I was very lucky. They actually embraced me, and they had all the genomics expertise, and I had the biological expertise. So we worked together and published this work, which at that point we sort of said that this was the atlas of blood dendritic cells and monocytes in human. And somehow that positioned me to be part of this massive global initiative, which was the Human Cell Atlas Initiative. And the inaugural meeting back in 2016, and I found myself being invited to that meeting at the Gibbs building in Wellcome. And the mission of this global initiative is to essentially create the equivalent of the Human Genome Project in terms of the full sequence of the human DNA, but in this case, the cellular constituents of the human body, and there are about 37 trillion cells. And that's what the Human Cell Atlas aspires to do, to create a reference map so that we can then benchmark against disease and perturbations. This is a global initiative. Anyone can join, so just go to that website. We now have many researchers from more than 80 countries as part of this initiative. So the premise is to use single-cell genomics or multi-omics technologies. The kind of like the earliest technology was sequencing RNA of individual cells, but the technology has really advanced, and you can now measure RNA and protein, RNA and open chromatin, and so on and so forth. So the technology that I've primarily used initially was the RNA sequencing, and now with surface protein and also open chromatin. And I'm going to show you some of the work that we've done leveraging these technologies. And what is the atlas going to look like? We outlined a roadmap for the developmental cell atlas, which is going to be the focus of the biology that I'll present today, and essentially emphasizing how a cross-disciplinary team approach, leveraging all of these different technologies, not just taking tissues and dissociating it to analyze individual cells, but also analyzing them in situ at single-cell resolution, at scale in terms of three-dimensional organs, and also small sections, and then integrating all of that information so we then have an integrated data set that we can query. And I'll show you how some of that data sets can be queried in a minute. So in order to achieve something like this, you have to have a slightly different approach to undertaking science. Traditionally, this is how science is generally done. It's kind of like very nimble, one person, one group, one project. But if you want to build something at scale, you really need many groups, many individuals working on one project, like the Human Cell Atlas. So this comes with its own challenges. You need the infrastructure to enable these types of consortia approach, the framework of how people can work together so that everybody's contribution is recognized, credit is duly acknowledged. How do you support positively team science? How do you have the best technologies? And how do you disseminate the data for the wider benefit of the research communities? So this is what I'm going to show you on the work that we've done to decode the developing human immune system. And I propose this as one of the areas that the UK could contribute to the Human Cell Atlas, partly because we have the Human Developmental Biology resource that has been around for 20 years. And there's a resource in Newcastle and London which actually supports studies into human development. So by using these kind of like high throughput profiling strategies and analyzing many, many organs from one embryo or fetus, you can begin to stitch together the information of how a distributed network like the immune system is differentiated and kind of populates the different organs. And something to highlight about the developing immune system, there's a component of space and time, in that you start off making blood and immune cells in the yolk sac, an extra embryonic structure, and then migrates to the liver and the bone marrow. And in liver and bone marrow, you have aortogonad mesonephros definitive hematopoietic stem cells that give rise to the blood and immune cells, compared to the yolk sac progenitor. But what is very distinct between the human and mouse is that you have bone marrow established at about 11 to 12 post-conception weeks, about the end of the first trimester. But you don't really get bone marrow hematopoiesis in the mouse until after birth. So how good a surrogate the mouse model is to human developmental hematopoiesis and immune system, remains to be fully established. So this is the work that we've done over the last few years. These are the publications that have come about. And the way that we've done this is to take all of the cells so that we can study all of the different lineages and in the environment that those lineages exist, specific organs. And that really allows you to see the emergent properties of the immune system. Immunology has been traditionally very focused on specific lineages. I mean, I grew up within the mononuclear phagocyte family, and you kind of see the world really from that perspective. But bringing the whole thing together allows you to see how the whole of the human's immune system is greater than the sum of its parts of the lineages. And also the need for interdisciplinary working, because this is a massive amount of work. You need a lot of expertise, not just about how blood and immune cells are formed, but how they function, understanding human development, and also the massive computational and bioinformatics input that is required to make sense of the data. And we're very keen to provide the data. This is a video, but it's not playing. Okay, great. So this is the data that is available through the web portal that my lab set up, and you can see here all of the data sets are actually available. You can click on it to download. This is just an example, and then it tells you the different types of data sets that are downloadable, and also all of the code and scripts you can see there, which are used in the analysis. And what this the accompanying web portal allows you to intuitively browse. So this is, each dot is a cell. You can browse or query the expression of the genes, and you can see here CD20 expression on the B lineage. This is the fetal liver, and you can also query other types of metadata associated with it. And more recently, we've incorporated the expression of genes that are known to cause inborn errors of immunity and also childhood leukemia. And you can then actually click on the data to query this, where there is a drop-down menu which actually allows you to look for diseases, and then a drop-down menu of the different types of diseases. Click on one, and all of the genes that are implicated and where they're expressed, in this case, the fetal bone marrow. So these are some of the ways that we've made the data intuitively browsable, so you can imagine as we scale up to the whole embryo and the whole human, the kind of information that anyone can get from those data sets. So what did we learn from this series of work? The first is that the blood and immune cells in human is established by 20 post-conception weeks. And that's the major site in the liver and also in the bone marrow. What is very interesting is granular poiesis really emerges in the fetal bone marrow. You don't have neutrophils in the fetal liver. It really becomes established in the fetal bone marrow alongside eosinophils and basophils. What is also striking is the prominence of B lymphopoiesis in the fetal bone marrow compared to the fetal liver, or there's no B cells, in fact, in the yolk sac. And you can see how the composition in the fetal bone marrow is primarily of B progenitors compared to the adult bone marrow, where the progenitor compartment is shrunken. And I'll come back to this later on, because one of the commonest types of leukemias in infant and childhood is B-cell leukemia. We put together all of the data sets and began to see more detail from that high resolution we got in that we were able to identify prenatal B1 cells. And if you remember, the kind of like innate lymphoid cells are the kind of like earlier versions of the adaptive T cells. The innate cells are much more ready to go. So the B cells have this equivalent of B1 cells. And you can see these cells here characterized by the expression of the genes, and we look to see for similarities of expression with the mouse B1 cells that have been more established. And also B1 cells are supposed to have high expression of IgM and also spontaneously secrete IgM. And when we FEC sorted these cells from the spleen, you can see here the CCR10 high B1 cells and the CCR10 low B1 cells are also the ones that are secreting spontaneously IgM, supporting or validating the findings of the B1 cells. And another exciting findings about unconventional T cells relates to how they may be selected, in that we know that T cells differentiate in the thymus, which we published about 2 years ago, detailing the cellular molecular profiles of those cells as they become double negative, double positive, and single positive T cells. We also showed unconventional and conventional T cells. Unconventional T cells are characterized by the expression of PLZF or ZBTB16. And what we found in the more recent analysis, including sequencing gamma delta VDJ sequences, is that unconventional T cells are more likely to have proximal combinations, whereas the conventional T cells have more distal VJ segments being kind of like recombined. And when we then took an in vitro culture model, an artificial thymic organoid model, to take iPSCs and differentiate them all the way to T cells, and then sequence them at different stages during that differentiation. And in this culture system, there are no thymic epithelial cells. The notch signaling is provided by the stromal cells that are expressing it. And what you find is that the vast majority of the T cells that you make, in fact, express ZBTB16 and are, in fact, more like the type 1 innate T cells. So this tells you how we can actually use the information from the in vitro and in vivo data to better engineer T cells as well. So the other interesting thing we observed about hematopoietic stem cells is how they change. They have intrinsic changes of HSCs within one organ, i.e. fetal liver, across gestational stage, in that the propensity of a hematopoietic stem cell to differentiate down different lineages is variable within one organ. So the earlier hematopoietic stem cells have a greater propensity to make erythroid cells, whereas the later hematopoietic stem cells from the fetal liver are more likely to make myeloid cells. And also the proliferative capacity of hematopoietic stem cells in the fetal liver is far higher than that in the fetal bone marrow. Also the data, because you're now looking at the non-immune cells, you can see how the niche supporting immune development is. And we identified different endothelial cell subsets that we then showed to line vessels in different regions of the fetal bone marrow, and how the fetal bone marrow niche is distinct from the liver niche, and a manuscript in preparation looking at the yolk sac niche. So those are some of the kind of insights, biological insights, from the analysis of the reference data set, the sort of healthy samples. And what we can now do is to use this to map chromosomal alterations and disease. And I'll show some examples of this now. So the first is looking at the chromosomal alterations from having a third copy of chromosome 21, or Down syndrome. And what you can then show here is how the blood and immune composition in the fetal bone marrow for the match non-Down syndrome fetal bone marrow compares, and what are the regulons that may be actually influencing this differentiation. So you can see here the expansion of the erythroid cells, the more abundant erythroid cells in the Down syndrome, and a contraction of megakaryocyte and B lineage in the Down syndrome fetal bone marrow. And this information can then be used to sort of understand why do you see the kind of like hematological malignancies associated with Down syndrome. Another example in a manuscript on bioRxiv, which is coming out next week, is using the reference fetal bone marrow data set to then map bulk RNA sequencing data of various infant leukemias. And what that allowed us to do is to say, what is the predominant malignant clone in these leukemias resembling in the fetal bone marrow data set? And you can see here for the cells in the red box, for those leukemias, they map to the early lymphoid progenitor rather than necessarily a more differentiated B cells. And these infant leukemias carry a much worse prognosis. And we generated site-seq data and looking at combinatorial protein expression of those malignant cells, which you can then now target with CAR T cell therapy. So adding, greater resolution to the diagnostic and prognostic and potentially treatment options for those patients. And the last example is that of developmental cell programs in inflammatory disease, and more specifically in inflammatory skin disease. So I was very interested to see whether our understanding of the developing immune system had any relevance to adult disease. And what we did then was to compare the developing skin with adult healthy skin and two of the common skin diseases, atopic dermatitis and psoriasis. And from that analysis, something really leapt out, which was one of the subsets of the vascular endothelial cells, VE3, on the left panel, was most resemblant transcriptionally to the endothelial cells that you find during development across several organs. And the same for macrophages. Macrophage 2 in adult skin was closest to the developing macrophages. And what was even more surprising was that VE3 and macrophage 2 were expanded in the lesional skin of atopic dermatitis and psoriasis, not just in our single-cell data set, but also the bulk RNA sequencing data set from repository data that we deconvoluted. And to cut a long story short, what we found was that the gene programs that were conserved was that about leukocyte recruitment and angiogenesis, and these VE3 in adult skin formed these kind of high endothelial venule-like cells in the skin where leukocytes were trafficking. And probing the data further, we distilled it down to interactions between ACKR1 and CXCL8 that was mediating this interaction, and the expression of those cells and their juxtaposition in the skin, in lesional skin, and how the V3 and MAC2 also declines, which I haven't shown here, following methotrexate treatment in a small cohort of atopic eczema patients. So this really boils down to the idea that, developmental cell programs can be co-opted in inflammatory skin disease, and what has been leveraged here is the leukocyte recruitment program used during development to seed immune cells in the tissue and also angiogenesis. What we don't know is whether the pathway is similarly regulated during development and in disease, and whether it has any other relevance for inflammatory disorders. Very briefly to end, in the sense that a lot of these kind of like framework, team science, allowed us to very rapidly deploy this to understand COVID-19, initially using the reference atlas to identify the expression of viral entry, receptor expression, and these were the first papers showing where ACE2 was expressed in all of the different human tissues, identifying the nose, the cornea as primary sites of potential entry, and also bringing together many researchers, many junior researchers working together very rapidly to then profile the peripheral blood immune response in COVID-19 as a surrogate to understand what's happening at the primary site of infection in the airway, and also the systemic response detected in the bone marrow. And I'm not going to dwell into this, but that the work has been published, and the ongoing work now is to try and incorporate our, this reference to kind of understand what goes on in a cohort of patients with end-stage renal failure, and also using electronic health records. All of the data can be visualized and downloaded at the COVID-19 Cell Atlas web portal. Just to kind of summarize now, that what I've shown is the clinical relevance of the Human Cell Atlas reference resource data sets, how it can be useful for tissue engineering cell therapy, how it can help us understand chromosomal alterations and childhood disorders, the fact that developmental pathways are used in adult pathology, and how this type of science and approach really puts us in a very good position to tackle some of the major challenges, including our recent pandemic, and how this is going to be the way future for precision medicine, particularly incorporating electronic health records and more robust data analysis. There's also public engagement associated with this work. Just Google One Cell at a Time, which is really interesting. I really enjoyed working with a group of artists and other poets, etc., for it. But I want to finish with 2 slides. One is the idea that we can do science differently, which allows lots of people to be acknowledged, to get the credit in a collaborative and supportive way. But really, this also needs equality and diversity. And I just want to focus a little bit on that diversity. It was International Women's Day a few days ago. And yesterday, we had a fantastic drinks reception, this amazing hall, the dining room, lots of pictures. You can see Queen Elizabeth here, Mary Robinson in the dining hall. And one of the things that strikes you is, how the kind of like, how not diverse some of these displays are, I guess. And perhaps this could be much improved, and how these corridors, how these halls are going to look in the future is really entirely up to all of us. I'm going to finish now to thank my lab for the fantastic science over the last few years. I've really enjoyed myself. The amazing collaborators around the world, and particularly working together with Sarah Teichmann at the Cellular Genetics program in Sanger, and our funders, and anyone who wants to join our lab, do get in touch with me. I'm very happy to talk. Thank you very much.
You talked about lessons from fetal development. Yesterday we were considering aging. I just wondered if there are things we can learn as we look at the aging immune system about disease.
That is a great question. Thank you very much. So I took out a slide, actually. One of the things that this allows us to do, and particularly with a lot of the other Human Cell Atlas initiative, is profiling pediatric, adult, and also aging samples. And now you can actually begin to really reconstruct what is going on, not just across developmental time periods, but all the way up to aging. And those are, great. There's going to be loads to be learned from that. Thank you.
It's a brave new world forming now with all kinds of new therapeutic targets and all kinds of ways of investigating what actually happens in vivo. How do you envisage that you can actually visualize and the effectiveness of drug therapy merging your technologies?
Good thought. Should I start? Yeah, I mean, this is really interesting in terms of, and it's a massive challenge. In some ways, what we can extrapolate now is confined by the technologies that are available. But what you find is that technology is constantly, moving forwards. And there's a lot of now whole organ imaging, which has been done on animal models, but also it's beginning to be adapted so that you can actually do it, in real time. So all of those are going to come into it. And alongside it, I think alongside the technological advances will be, critical computational advances to allow us to begin to stitch information that you get from another modality with added, integrating modalities, basically. That's going to be a computational challenge, and that's going to kind of like go hand in hand with this. And so this is sort of like forward thinking. We can do it to some extent now, but it's just going to get much better, and eventually we will get to that stage where we'll be able to sort of like put all of this information together and probably have less reliance on animal models. That's my gut feeling.